Biochemistry classes. Today I am back with a video on glycogenolysis. What is glycogenolysis? It is a process or it is a metabolic process involving the breakdown of glycogen to glucose. Where does it occur? It occurs in the cytoplasm of the hepatocytes when in fasting condition and in exercising muscles. It is regulated by glucagon and insulin in case of liver, epinephrine in liver and muscle, and calcium in case of muscle. The key point to remember is it is not the simple reversal of glycogenesis. The end product of glycogenolysis in liver is glucose and in muscle it is glucose 6-phosphate. This is a common catch question in Viva table that why the end product of glycogenolysis in muscle is glucose 6-phosphate. The answer is the muscle lacks the enzyme glucose 6-phosphatase. Now let us look at the steps of glycogenolysis. Step 1. What does it involve? It involves shortening of the glycogen chain by glycogen phosphorylase. Glycogen phosphorylase is the key enzyme for glycogenolysis. Now, this glycogen, this is a polysaccharide involving glucose residues. It has both linear chain and branching chain. In linear chain, the glucose residues are linked by alpha 1,4 glycosidic linkage, and in the branching point it is linked by alpha 1,6 glycosidic linkage. Now, when the glycogen phosphorylase acts upon the glycogen, one molecule of inorganic phosphate is added and glycogen is shortened by one glucose residues. If there is n glucose residues, the product will have that glycogen that is formed will have n-1 glucose residues and glucose phosphate is formed as an end product. The glycogen phosphorylase, the key enzyme of this step, requires pyridoxal phosphate as cofactor. Here pyridoxal phosphate acts as cofactor, not coenzyme, and this step is also known as phosphorolysis, and we will have shortening of the glycogen chain. The glycogen phosphorylase will start acting from the non-reducing end of the glycogen and it will start trimming the glycogen molecule until and unless there are 4 glucose residues from the branching point. So, you can see that there are 4 glucose residues from the branching point on the other side. Either side we have the 4 glucose residues. So, step 1, that is phosphorolysis, which involves shortening of the glycogen chain by glycogen phosphorylase, and the end product is glucose phosphate. Glycogen phosphorylase requires pyridoxal phosphate as the cofactor. Now, let us see what happens in step 2. And this product which is formed by the action of the glycogen phosphorylase, this branch product is known as limit dextrin. Glycogen phosphorylase further cannot act on this limit dextrin. It involves removal of branches by debranching enzyme. Now, this debranching enzyme is a bifunctional enzyme. That means it is an enzyme. So, by default it will be protein in nature and the domains of the enzyme will serve 2 catalytic activities. Let us check what are those. In the first step, what will happen? The debranching enzyme will do the action of alpha 1,4 transferase activity. That means it will transfer the glucose residues that are linked in the branching chain or in the branch chain 3 glucose residues to the non-reducing end, and they will be linked by alpha 1,4 linkage. So, here we can see that only 3 glucose residues, they are transferred to the linear chain, and only 1 glucose residue is present at the branching point. So, this is the first activity of the debranching enzyme. Now, second part that debranching enzyme will do, that is alpha 1,6 glucosidase activity. That means it will act at the alpha 1,6 glucosidic linkage which is present at the branching point, and there will be removal of 1 glucose residue. So, we will get a debranched glycogen. So, in the step 2, two actions take place together. First one is alpha 1,4,1,4 transferase activity, which will act on the branched point, remove 3 glucose residue to the linear chain, and second part is the alpha 1,6 glucosidase activity, which will act on the alpha 1,6 glucosidic linkage at the branch point and remove the glucose residue. Formation of glucose 6-phosphate and glucose. This deep branch glycogen will again be acted upon by the enzyme glycogen phosphorylase, and glucose 1-phosphate will be formed. Now, this glucose 1-phosphate, it will again be isomerized by reversible reaction to glucose 6-phosphate, and phosphoglucomutase is the enzyme. Now, this glucose 6-phosphate is the end product of glycogenolysis in the muscle, and in the muscle this glucose 6-phosphate will enter the glycolytic pathway where it will be converted to pyruvate. Then this pyruvate will go to transamination and it will be converted to alanine, and it will further go to the glucose alanine cycle. Now, this glucose 6-phosphate, what will happen in the liver? In the liver, it will enter the endoplasmic reticulum. How it will enter the endoplasmic reticulum? By another transporter or with having catalytic property, that is glucose 6-phosphate translocase. And in the endoplasmic reticulum, it will be acted upon by the glucose 6-phosphatase enzyme, and it will be converted to free glucose. So, this part of the reaction, it will take place in cases of it will take place only in cases of liver. This part will take place in the liver, but in case of muscle, the end product will be glucose 6-phosphate. Now, this is the major metabolic fate of glycogen in the liver or in the muscle cytoplasm. 1 to 3% of the glycogen that is acted upon by the lysosomal enzyme. What is that? Alpha 1,4 glucosidase, that is known as acid maltase. And if there is any deficiency of this enzyme, this will lead to Pompe's disease, that is again categorized as type 2 glycogen storage disease. So, majority of the glycogen is converted to glucose 6-phosphate in cases of exercising muscle, and it is converted to free glucose in cases of liver. However, a small percentage of the glycogen can enter the lysosome, and it is acted upon by alpha 1,4 glucosidase, and it is converted by acid maltase. If there is any inherited deficiency of this enzyme, that leads to type 2 glycogen regulation disorder or glycogenolysis. We know every metabolic pathway have two types of regulation: allosteric regulation and hormonal regulation. So, in cases of liver, hepatic phosphorylase or hepatic glycogen phosphorylase, it is activated by low glucose. If the body glucose reserve is low, then it will give signal for further breakdown of glycogen, and it is inhibited by increased glucose. That means in cases of well-fed state, it is inhibited by ATP and it is inhibited by glucose 6-phosphate. In cases of muscle phosphorylase, the allosteric activator is AMP and allosteric inhibitor is ATP and glucose 6-phosphate Remember one thing, free glucose has no allosteric regulation on muscle phosphorylase. Muscle phosphorylase is not having any coordination with free glucose concentration. Now coming to hormonal regulation. Hormonal regulation, it is mainly taken care of via the insulin and glucagon in cases of liver, via epinephrine and in cases of both liver and muscle, and this hormonal regulation, that is via the covalent modification of the key regulatory enzyme we know, phosphorylation and dephosphorylation. Now for this we should remember three cardinal points. First one is, what does glycogenolysis or what does glycogen phosphorylase do? It will increase glucose concentration, right? So which hormone will be responsible for increasing the glucose concentration in the body? It is glucagon. So glucagon will activate the glycogen phosphorylase. Clear? Third point is, how does glucagon activate? Glucagon activates by phosphorylation. Glucagon activates by phosphorylation. So what is the cardinal points that we have studied regarding the regulation of glycogenolysis? First point is hormonal regulation. It is done by covalent modification. What are the cardinal features of covalent modification? Phosphorylation and dephosphorylation. How does this phosphorylation and dephosphorylation occurs? Phosphorylation occurs via the glucagon. Next is, glycogen phosphorylase is active in phosphorylated form. So these are the three important features whenever you are going through a metabolic pathway, remember. Now glucagon has no role on muscle glycogenolysis. Glucagon has no role on muscle glycogenolysis. Now let us check what happens. Glucagon or epinephrine, they will act via the serpentine G protein coupled receptors. So this red marked oval is the glucagon or epinephrine that will act on the G protein coupled receptor, and this, once they will act on the G protein coupled receptor, they will activate the membrane bound enzyme, that is adenylate cyclase or adenylate cyclase. It will activate the enzyme. What this enzyme will do? This enzyme will act on adenosine triphosphate and will break it down to cyclic AMP. Now this cyclic AMP will activate the protein kinase A to its activated form, and this activated protein kinase A, it will act on glycogen phosphorylase kinase B. This small b is for the inactive form, and it will be phosphorylated to glycogen phosphorylase kinase A. It will be phosphorylated and it will be thus activated. Now this glycogen phosphorylase kinase, what it will do? It will activate the key regulatory enzyme glycogen phosphorylase to its active form. Now what will happen in well-fed state? As I have already said that glycogenolysis takes place when the body glucose reserve is low, and within 12 to 18 hours of complete fasting, the entire glycogen reserve of the body is exhausted. Then we have to depend on gluconeogenesis. So what will happen? In well-fed state, there is increased insulin in the body. This insulin will act on protein phosphatase 1. And what this protein phosphatase 1 will do? It will act on the glycogen phosphorylase, phosphorylated form and dephosphorylated. So it will be deactivated. It will be deactivated. Moreover, this cyclic AMP, this is the key regulatory second messenger that will activate the protein kinase A. So if there is well amount of glucose in the body, insulin is the main hormone. So this insulin will activate the phosphodiesterase and it will break down cyclic AMP to 5' AMP. So this is how insulin and glucagon acts in a counter regulatory way. So via cyclic AMP dependent protein kinase A, glycogen phosphorylase kinase will be activated, and this will in turn activate glycogen phosphorylase to its active form. Whereas when the body glucose reserve is low, the insulin will be more and it will activate the protein phosphatase 1 that will dephosphorylate the glycogen phosphorylase, and it will also activate the phosphodiesterase to 5' AMP, to 5' AMP. So these are the important regulation of. So this is how glycogenolysis is regulated. Just the reversal type of regulation takes place in cases of glycogenesis, that is the key regulatory enzyme of glycogenesis, that is glycogen synthase, that is active in dephosphorylated form. If you want to check the video, the link is given in the description box or in the I button above. Now, as I have said that glycogenolysis also takes place in exercising muscle, and here it is exclusively regulated by the calcium. Calcium is a key point in regulation of muscle glycogenolysis. So let us check how does it happen. In exercising muscle, what happens? The nerve impulses causes membrane depolarization, and this membrane depolarization of the endoplasmic reticulum, where in cases of muscle it should be called a sarcoplasmic reticulum, where the calcium is stored, the calcium is released and it binds to the calmodulin. Now this calcium calmodulin complex, it will bind to the muscle phosphorylase kinase and it will be activated. There are 4 subunits of muscle phosphorylase: alpha, beta, gamma and delta, and it binds to the muscle phosphorylase kinase, this calcium calmodulin complex, and it will be activated, which in turn will activate the muscle phosphorylase, Now what happens in cases of liver? In cases of liver, the epinephrine, it will again cause the activation of the endoplasmic reticulum and the release of calcium calmodulin complex in a similar way, and this calcium calmodulin sensitive phosphorylase kinase will be activated. Here it is in cases of muscle, it is muscle phosphorylase kinase, and in cases of calcium, it is calcium calmodulin sensitive phosphorylase kinase, which in turn will activate the hepatic phosphorylase. So this is how calcium plays a key regulatory role in cases of regulation of glycogenolysis in cases of muscle. So to sum up, glycogenolysis is the metabolic breakdown of glycogen to glucose. Where does it take place? In the cytoplasm of the liver and the exercising muscle. It is not a simple reversal of glycogenesis. The key regulatory enzyme of glycogenolysis is glycogen phosphorylase. What are the other enzymes involved? The debranching enzyme, which is a bifunctional enzyme, and the glucose 6-phosphatase, which is exclusively absent in the muscle. The end product of glycogenolysis in liver is free glucose. In muscle, it is glucose 6-phosphatase. It is allosterically regulated by glucose exclusively in liver, and ATP and glucose 6-phosphate are inhibitors both in cases of muscle and liver, and AMP is an allosteric activator in cases of muscle. Hormonal regulation, it takes via the insulin and glucagon in cases of liver, via the epinephrine in cases of liver, and exclusive regulation via calcium in cases of muscle. If you like my channel, then keep watching, and thank you for watching. Keep watching.